Dear students, welcome to BIORATNA. Today we are going to do one shot on photosynthesis. How easy it is for us now to say that photosynthesis is the process of the formation of an organic molecule, that is glucose, from inorganic molecules like CO2 and H2O in presence of chlorophyll and sunlight. In presence of chlorophyll and sunlight. Sun, the one who is present there. We organisms, we are present here on the earth to find out that connection. That it is the sun, it is the solar energy which is providing this food to the organisms on the earth. The green plants, they are capturing the solar energy and now they are doing photosynthesis, and the whole biosphere, all the living organisms are dependent on that food. So you can understand how difficult, step by step, so many contribution of so many scientists to find out the intricacies of the process of photosynthesis. Let us first start with the contribution of the scientist Van Helmont. What he did, he grew a willow plant. He grew a willow plant in a weighed amount of soil. That means the soil was also weighed, and then he put the plant into it. And after 5 years, he weighed the plant again. What he found out? He found out that the weight of the plant was approximately about 74 kg more than its previous weight. जब plant लगाया था, छोटा सा plant था, उससे around 74-75 kg more weight it was having. Now he had previously weighed the soil also. So now he weighed the soil and he found that the decrease in the weight of the soil was negligible. I repeat, the decrease in the weight of soil was negligible. So he said, what was his conclusion? He said that plant is all water. Plant is all water. This was his observation. You cannot ridicule it. He was correct when he was saying, he was stating his observation. He said that the weight plant has not gained from the soil. Soil is approximately means about the same in its weight. Only water I was putting there. So all the extra weight has to come from water. So the all plant is simply water. Was it totally wrong? Was his experiment of not any significance? No, not so. Till Van Helmont, it was thought that it is the soil from which the plants are getting all its food, all its nutrients. There was no concept of photosynthesis. इसने भी photosynthesis खोज के नहीं निकाला. But now he proved कि no, this belief that all the nutrients come from soil is wrong and we have to look further. He said it is water. That was his observation. It is simply our observation. It is our understanding which goes on changing. Let us see how. Now come to this another scientist. This is Joseph Priestley. What he was doing, he took a mouse inside a bell jar. This mouse, he dies after some time because he is continuously respiring. He is taking the oxygen from the air. It was not known at that time, na? So when he is taking in the oxygen he has taken in and he has released so much carbon dioxide and he dies. He repeats the experiment with a candle. The candle is burning. The same thing happens after some time. That flame also extinguishes. Now he puts a green plant, potted plant also inside that bell jar together with the mouse and the burning candle. Mouse doing respiration, candle it is burning. But now the mouse also leaves and the candle also keeps burning. So naturally, what is the conclusion? So Priestley says that the respiration and the burning, what it does? It removes something from the air. Removes something very important for the living from air. They remove it, but plants, the green plants replenish it. ये उसे वापस कर देता है. Fine. अभी भी vague है. Still it is vague. Now come to the next experiment, that is by Jan Ingenhouz. Jan Ingenhouz. What he does, he came across Priestley's experiments. He does it a different setup where he uses one aquatic plant. Once he places it in sunlight and once in dark. What he finds out, that when the plant is kept in sunlight, at that time the bubbles, it comes out from the green parts of the plant. But when the setup is kept in the dark, at that time the bubbles, they stop coming out, and this air bubbles were found out to be oxygen. Thus, what the most important finding of Jan Ingenhouz is that he now connected photosynthesis with sunlight. Now for this process, अभी भी photosynthesis समझ में नहीं आया है, but now he has said that this process is to be related, has some direct connection with the sun. So the process is related to sunlight. One very great finding. And the bubbles which are coming out, it was oxygen. Next one is von Sachs. His findings you will remember easily by thinking of his name as Sachs, S-A-C, Sachs. What were his findings? Von Sachs, he found that when the plants grow, then they form glucose. Plants, they form glucose, and this glucose gets stored in the form of starch. This is one thing, the storing of the glucose. And the next is that the green parts of the plant, where all these things are going on, the different scientists are little by little finding out something. So these green parts, they consist of a pigment called chlorophyll inside chloroplast. Inside chloroplast. So you remember these two things, glucose and chlorophyll. This is the finding of von Sachs. Now there is one novel experiment by T.W.Engelman. Very nice experiment. What he thinks, that the sunlight which has something to do with the process, and the light can be divided into 7, that V, I, B, G, Y, O, R. Fine. So out of these 7, which one helps in photosynthesis? This is one question. And then this chlorophyll molecule, chlorophyll is also of so many types. Na? Chlorophyll A hai, B hai, C hai, D hai, and then there are other pigments also in the plant. So which of is responsible for this process? And from this Weber, which wavelength is active during photosynthesis? Two things, and this is the which pigment and which wavelength of light. two questions. How he designs his experiment. He splits with the help of prism. He splits the sunlight. मिल गया spectrum. That is one part done. VIBGYOR. Now he has got. Now he needs one plant. The plant which is going to do the photosynthesis. He used the algae cladophora. He uses the algae cladophora. Now he allows, now this cladophora, it is placed in water. From the prism, this split ray is allowed to fall on cladophora and it starts photosynthesis. कहाँ पे हो रहा है ये photosynthesis? कैसे पहचाने? For that, what he does, he uses radioactive aerobic bacteria. You are understanding what he is doing? Aerobic bacteria. Now, when the 7 different wavelengths, they fall on the cladophora, then these aerobic bacteria, they accumulate at the blue and red light zone, blue and red wavelength, where the oxygen is, photosynthesis जहाँ होगा वहाँ पे oxygen निकलेगा. So at that point, all these aerobic bacteria, they accumulate in these two zones, blue and red, showing that photosynthesis is active at this point, and this is called the action spectrum. This is the action spectrum of photosynthesis. Action, कहाँ action हो रहा है? Blue और red zone में action हो रहा है. Wonderful experiment. Now by mid-19th century, we thought that we understood the steps of photosynthesis very properly, and the equation which we gave, it was CO2 plus H2O is equal to CH2O. This is reacting with this, and then this oxygen, it gets released. When it is glucose, then this automatically becomes C6H12O6. Fine. Now comes here, of course, the light is there and the chlorophyll is also there. Now comes O Niel. What he says? He says that for the autotropism, for the production of glucose, water is not a must. It is not that it is only water which can do this. Actually, the plants, they need, do listen carefully, the plants, they need a hydrogen donor. Plants, they need a hydrogen donor which can give its hydrogen to the carbon dioxide, thereby reducing the carbon dioxide into glucose. And in this process, it gets oxidized. Here the water molecule, it is getting oxidized into oxygen. He gives the example of the purple and green bacteria. This is the purple and green bacteria, where this is CO2 plus H2S, not H2O. Then this is CH2O plus S. Here it is not oxygen which is given out, but it is the sulfur which is given out. So we again have to rethink. यहाँ देखो बच्चों, यहाँ हमने क्या सोचा था? यहाँ हमने सोचा था कि this oxygen, it is released from carbon dioxide. लेकिन जहाँ हम H2O के बदले H2S यूज करते हैं, then it is not oxygen which is released, but it is the sulfur which is released. Which means sulfur कहाँ से आया? H2S से आया, CO2 से नहीं आया. So this oxygen is not coming from CO2 but water. This was also proved by using radioactivity. Now we modified this equation. We modified it into 6 CO2 plus 12 H2O is equal to C6H12O6 plus 6O2 plus 6H2O, ताकि हम दिखा सके कि the oxygen which is released, it is coming from water. The step by step we have understood something which was very difficult to conceive, but all these scientists, our science, it helped us to understand photosynthesis. Now this photosynthesis, that process by which the solar energy is trapped by the plants by means of their pigments. They have some pigments which are placed inside an organelle called chloroplast. So let us see what is this chloroplast and what are these different types of pigments. This is the structure of a chloroplast. A chloroplast has a discoid shape. This is its outer membrane. This is its inner membrane. These are the two membranes of chloroplast. Inside, this is stroma. Inside this is stroma, and the most important thing, these are, inside you will find many coin-like structures stacked upon one upon another. So these are different coin-like structures which are present in the stroma. If you take a single of this coin, then this is called thylakoid. Only one coin is thylakoid, whereas if you take the whole stack of the coins, पूरा का पूरा कॉइन का स्टॉक ले लेते हो बच्चों, स्टैक ले लेते हो बच्चों, then this is grana. पूरा लेते हो उसका जो स्पेलिंग है वो छोटा है और जो सिंगल लेते हो उसका स्पेलिंग बड़ा है, थाइलाकॉइड. शुरू में इसी तरह से कुछ लिंक कर करके याद किया करो. एंड देन these grana, they are interconnected. तो ये इस तरह से कनेक्टेड होता है and these are called lamellae. And as these lamellae are present in the stroma, so you can call it stroma lamellae. And as these are connecting to grana, that's why you call it intergranal lamellae. Intergranal lamellae. Either stroma lamellae or intergranal lamellae. Now, other than this structure, you will also find small, these structures, these are the ribosomes. These are ribosomes. Then you will also find some bigger granules, which are the lipid droplets. These are lipid droplets. And one important thing, the glucose which will be formed here, it will be converted into starch. So these are the starch molecules. Here you can see one, here I will draw one, another, here another. So these are the starch molecules. So this is the structure of chloroplast. Now the most important thing which you want to know, that is, in the process of photosynthesis, first the solar energy is trapped by the plants. This is one very distinct process. Means photosynthesis can be divided into two sub-processes. Na? One is simply the trapping of energy. Solar energy को trap कर लिया. Fine. Energy जब मिल गई, then that energy is used to prepare the food. So this trapping of energy is called light reaction and the formation of food is called dark reaction. I repeat. जब सोलर सन के प्रेजेंस में सन की लाइट पिगमेंट्स एब्जॉर्ब करते हैं और एटीपी और NADPH बनाते हैं, then this is called light reaction. This is light reaction. And Next is dark reaction. What is dark reaction? Dark reaction does not take place in dark. ये अंधेरे में नहीं होता. It simply means it is not trapping the solar energy. It is not trapping solar energy. It is preparing the food after the trapping of energy has been completed by the light reaction. And don't think that dark reaction does not depend on light. Dark reaction does not take place in dark, but dark reaction is indirectly dependent on light. कैसे? वो सोलर एनर्जी को ट्रैप करते हुए ATP NADPH नहीं बना रहा है, but its whole mechanism, for its food preparation, it needs the ATP and NADPH. These are prepared during light reaction, so it is indirectly dependent on light. So it is also taking place in light only. जैसे सूरज महाराज आ गए, then light reaction starts and the ATP and NADPH बन गए. जैसे ATP and NADPH बन गए, immediately the dark reaction starts. Clear? So dark reaction, remember, dark reaction does not take place in dark. Both are going on in light. First, the light reaction starts because dark reaction needs energy. That energy is formed during light reaction. This does not mean that dark reaction is not dependent on light. It is indirectly dependent on light, as it is dependent on ATP and NADPH produced during light reaction. Fine? You need to know, किसी भी ऑर्गेनिज्म में, लिविंग ऑर्गेनिज्म में, ऑल रिएक्शंस आर कैटालाइज्ड, एंजाइम्स की मदद से होते हैं. सो लाइट रिएक्शन वाले जो एंजाइम्स है वो कहाँ प्रेजेंट है और डार्क रिएक्शन वाले कहाँ प्रेजेंट है. वेरी इंपॉर्टेंट, वेरी वेरी इंपॉर्टेंट. ये दो आपके क्लियर कर लीजिए कि पहले लाइट रिएक्शन स्टार्ट हुआ. सपोज यहाँ से ऊपर से होते हुए ये लाइट आ रही है, ठीक है, ऐसे याद रख सकते हो. ऐसे लाइट आ रही है, पहले उसे कौन मिला? पहले जैसे वो एंटर करता है तो उसे स्ट्रोमा मिलेगा ना. तो स्ट्रोमा में लाइट रिएक्शन स्टार्ट कर सकता था, नहीं करता है, वहाँ एंजाइम्स ही नहीं है लाइट रिएक्शन के. सो नाउ इट मूव्स टू द ग्राना सिस्टम, membrane system I should say. This membrane system, it consists of the grana and this lamella. Grana and lamellae. This grana and the lamellae which are joining the two grani. Here the enzymes for light reaction is present. Enzyme for light reaction. Fine. अंदर आके उसे मेहनत करके यहाँ तक अंदर आया और फिर इन लैमेली में और ग्राना में उसे एंजाइम्स मिल गए. सो द लाइट रिएक्शन इज टेकिंग प्लेस इन दिस थैलाकॉइड्स एंड द स्ट्रोमा लैमेली. And then dark reaction enzymes, they are present in stroma. ये जो stroma है, here the dark reaction वाले enzymes present है. So dark reaction takes place in stroma, light reaction takes place in grana and the lamellae. Clear? आगे बढ़े. Now this chloroplast. This chloroplast, it is not just present in leaves, it is also present in all green parts of the plant. जहाँ भी plant green है, there these chloroplasts are present. These chloroplasts are present with their pigment. The mesophyll cells, the mesophyll cell का mention है आपके NCERT में कि the mesophyll cells, they contain large number of chloroplast. तो ये मेसोफिल सेल आपको याद है, डायग्राम आपको याद है, 10th में पढ़ा था आपने. तो अगर आप लीफ का सेक्शन लेते हो, इफ दिस इज द अपर एपिडर्मिस एंड देन हियर देयर आर सम टाइटली पैक्ड सेल्स. व्हाट आर दिस सेल्स? दीज आर द पैलिसेड सेल्स. दीज आर पैलिसेड सेल्स. देन अगेन अनदर लेयर ऑफ पैलिसेड सेल्स. Then there is no gap between these palisade cells. Then you will find these cells called the spongy cells. These are the spongy cells and they have this intercellular space filled with air in between them. तो उनमें यहाँ पे गैप वाले होते हैं, and this palisade and this spongy taken together, it is called mesophyll cell. This is mesophyll cells. यहाँ नीचे में और एक लोअर एपिडर्मिस है, यहाँ पे नीचे में और लोअर एपिडर्मिस है, and this is your diagram of section of the leaf. तो ये जो palisade है, ये palisade cell, this palisade cell and this spongy cell together they are called the mesophyll cells. और ये palisade cell में यहाँ big vacuole होगा, यहाँ पे एक nucleus होगा, and there are these structures in the cytoplasm. These are the what? This chloroplast. Fine. Now this chloroplast, they have pigments. What are pigments? Pigments, they are the molecules which can absorb the sunlight at different wavelengths. Means जो pigment कौन सा wavelength catch कर सकता है, ये fix है. And what are these different pigments? So important. Those pigments which are capturing the solar energy, and then the cell will convert it into chemical energy, ATP and NADPH. So these pigments are, one is chlorophyll A, then it is chlorophyll B, then xanthophyll and carotene. So this chlorophyll A is blue-green. This is blue-green. Chlorophyll B, it is yellow-green. This is yellow-green. Then xanthophyll is yellow and carotene is yellow to yellow-orange. This is yellow-orange. These are 4 different types of pigments. Chlorophyll A is called main pigment and rest 3, these are called accessory pigments. Why so? Let us see. These are the accessory pigments. Chlorophyll A, this is the main pigment. So here I am drawing the action spectrum of photosynthesis. This one, and then this is at blue and this is at red. VIBGYOR, ये B, blue और red region में maximum photosynthesis होता है. The action is of photosynthesis is most active in blue and red region that you have seen in that experiment ना, क्लैडोफोरा वाला. किसने किया था? इंगलमैन ने किया था. क्लैडोफोरा रखा प्रिज्म से, he split the ray into the 7 components via BGYOR, and then he found by means of aerobic bacteria that they assembled at the blue and red zone where maximum oxygen was released. Now blue और red zone में तो हो रहा है. अब हम अगर ये देखते हैं, this is action, most action is going on in blue and red region. अब हम अगर इन चारों का एब्जॉर्प्शन स्पेक्ट्रम देख ले कि अगर क्लोरोफिल ए ले लेते हैं तो क्या ये भी एब्जॉर्ब ब्लू और रेड रीजन में ज़्यादा करता है? Then it will be the photosynthetic pigment. So when we see the absorption spectrum of chlorophyll A, then we see कि ये ऐसे and then बीच वाले BGYO ऐसे अवॉइड करते हुए then it moves upwards, और ये है हमारा this blue one, this is the absorption spectrum. This is absorption spectrum of chlorophyll A. And what is this? This is the action spectrum. This is action spectrum. Fine. Okay. So when we watch this, then we see that the chlorophyll A is the major pigment for photosynthesis. Of course, if you observe it carefully, you will see that the peaks, they do not overlap. वन टू वन ओवरलैपिंग इज नॉट देयर. यहाँ देखो ये इस पोजीशन में इसका पीक है, और यहाँ देखो ये क्लोरोफिल ए का यहाँ पे है, थोड़ा सा हटके है. व्हाई सो? बिकॉज दिस क्लोरोफिल ए is absorbing the light at a particular wavelength, or the accessory pigments, that is chlorophyll B, then xanthophyll, then carotene, these are also absorbing light at different wavelengths, and they are shifting it to the chlorophyll A. They are also passing their energy to chlorophyll A. So they also have great role. They are enabling the light energy at different wavelength other than the chlorophyll A can absorb, to be utilized for photosynthesis. At the same time, they also protect chlorophyll A from photooxidation. Now let us study the light reaction. What are the different steps of light reaction? First is, of course, the absorption of solar energy. Then there is splitting of water. So when water is split, oxygen will be released, and that oxygen is a waste matter for the plant, so it is given out. And most importantly, light reaction is for the production of ATP and NADPH. So ATP and NADPH molecule is formed. How? The question is how? How the pigment molecules are helping in this? Now understand what is LHC. LHC, that is light harvesting complex. Light harvesting complex means the different types of molecules together with the proteins which are absorbing the light of different wavelengths. These are arranged under PS1 and PS2. PS1 and PS2. In PS1 and PS2, this diagram will be helpful in understanding this, that these are the antenna molecules. Antenna molecules are what? Antenna molecules are molecules other than chlorophyll A. The pigment molecules other than chlorophyll A, like chlorophyll B, carotene, xanthophyll, what they are doing? They are absorbing the solar energy, इन सबको जो मिल रही है, and they all then pass it to, all of them pass it to a reaction center. This is antenna molecule and this is the reaction center. Reaction center. A reaction center is always chlorophyll A. Reaction center is chlorophyll A, and these rest antenna molecules are chlorophyll B, carotene and xanthophyll. When all these antenna molecules transfer their energy to chlorophyll A, then this chlorophyll A molecule, it loses its electron. It loses its electron due to higher energy level. It comes out of its orbit and then it is accepted by primary acceptor. This is primary acceptor. Fine. So this is a photosystem. This is a photosystem with its antenna molecule, with its reaction center. Then what is the difference between PS1 and PS2? In case of PS1, this chlorophyll A, the reaction center, it has absorption maxima at 700 nanometer. Whereas in case of PS2, the chlorophyll A molecule has absorption maxima at 680 nanometer. 680 for PS2 and 700 nanometer for PS1. तो ये सारे मॉलिक्यूल्स हैं जो सारे मिलके अपनी डिफरेंट वेवलेंथ वाली सोलर एनर्जी को कैप्चर करते हुए उसे अपने रिएक्शन सेंटर दैट इज क्लोरोफिल ए को ट्रांसफर कर रहे हैं. सो chlorophyll A is always the reaction center and the rest of the molecules, these are the antenna molecules. This PS1 and this PS2, these two photosystems are going to take part in the light reaction. We are seeing the different components which are taking part in the light reaction. When we see the light reaction, then we will see that there are two different ways in which ATP is formed. ATP and NADPH is formed. In one, both of these take part, PS2 as well as PS1, whereas in the another, only PS1 takes part. And the one called Z photophosphorylation, where both of them take place, then you will see that this 1 and 2, this number is not given as per the sequence in which they start the reaction. Actually, it is PS2 which starts the reaction, then comes PS1. So, let us see how this ATP and NADPH are formed by two different reactions. First, we will take the non-cyclic or Z photophosphorylation. Phosphorylation means that type of reaction in which we are going to form the energy-rich compound ATP. First one, it is non-cyclic photophosphorylation. This is non-cyclic photophosphorylation. This is also called Z-scheme. Why Z-scheme? Let us see. So, non-cyclic means what? Non-cyclic means the electrons which are released by one system after completing its work, it does not come, its work means formation of ATP. It does not come back to its original photosystem. Then it is called non-cyclic. I repeat. One photosystem, it absorbs the light energy. Now its reaction center, it releases electrons. This electron passes through many carriers and then it produces this energy rich compounds ATP and NADPH, but it does not come back to the original photosystem. Then it is called non-cyclic. How? This one starts with photosystem 2. So this is photosystem 2. This is its LHC, light harvesting complex. This is the solar radiation. Now the reaction center, after absorbing the wavelength of 680 nanometer, it now releases its two electrons. The electrons, they leave their orbit due to this high energy and this is released and it is accepted by a primary acceptor. This is photosystem 2. This is photosystem 2. This acts at 680 nanometer. 680 nanometer. Now what happens to this electron? This electron passes through a chain of molecules. This process is called electron transport system, and the chain of molecules through which it passes, they are the carriers. जैसे एक रिले रेस चल रही है, सारे एक के बाद एक उस इलेक्ट्रॉन को एक्सेप्ट कर रहे हैं and they are the cytochromes. What are they? They are cytochromes. Now the electron passes through them and in this process here the ATP is formed. Here ATP is formed and then this electron, it gets transferred to PS1. PS1 का ये है LHC. इससे थोड़ा हाइयर दिखाना पड़ेगा बच्चों, because this is at 700 nanometer. So this is the LHC of PS1. It has got two electrons from PS2. it has got 2 electrons, but it receives the solar energy. It also loses its electrons. So when it loses its electron, it has lost its electron, but it has gained electron from another photosystem, that is PS2. Its 2 electrons, it is accepted by a primary acceptor. This is photosystem 1. This is photosystem 1. Now from here, this electron, it is accepted by NADP, so that it gets converted into NADPH plus H plus. This is one energy rich molecule and this is one energy rich molecule. So we get ATP and NADPH. What we have seen, PS2 and PS1. This is PS2, this is PS1. Both of them have lost their electrons, but the electron from PS2, after giving out the ATP, after the formation of ATP, it is donated to PS1. Electron of PS1, now it helps in the formation of NADPH. So at the end of this reaction, you see that this one, PS2, it is short of its LHC, it is short of 2 इलेक्ट्रॉन. यहाँ 2 इलेक्ट्रॉन कम है, इसके 2 इलेक्ट्रॉन चाहिए. तो ये इलेक्ट्रॉन आएंगे कहाँ से? ये सिस्टम तो कंप्लीट हो गया, एटीपी बना दिया इसने, एनएडीपीएच बना दिया, लेकिन फिर से ये काम नहीं कर पाएगा अगर इसके इलेक्ट्रॉन इसे नहीं मिले. फॉर दिस पर्पस द फोटोलाइसिस ऑफ वाटर टेक्स प्लेस. अब वाटर से क्या होगा? दिस वाटर, it is split and then it forms 2H plus 2E minus and half of O2. And these electrons, then it gets its electron from the splitting of water, and that oxygen, it is a waste product, it is released in the atmosphere. Thus PS2, it also gets back electron, but it gets it from water. It is not its original electron. Its electron is given to PS1. One thing. This water splitting complex, it is attached to the PS2. Naturally PS2 को ही चाहिए, तो PS2 के साथ ही attach होगा. So the water splitting complex is attached to this PS2, and this PS2, it is present. This is one thylakoid. This PS2, it is present on the inner surface of the thylakoid. This is one more hidden fact which you should be careful about. Ki this water splitting complex attached to PS2 present on the inner side of the thylakoid. Now let us see one more type of photophosphorylation. Oh, why it is called Z scheme? See, this looks like a Z. This, then here, and then here. So it looks like a Z scheme. That's why it is called Z scheme. There is one more different type of photophosphorylation, which is cyclic. In it, only PS1 takes part, and the electron which is lost by PS1 comes back to the PS1 itself. उसी को वापस मिल जाता है, and only ATP is formed in cyclic photophosphorylation. The cyclic photophosphorylation. We have already seen the non-cyclic photophosphorylation. You know the meaning of non-cyclic one? Means electrons released from the source do not return back to the source, and that source was PS2. Non-cyclic one, it started with not PS1 but PS2. Then its electron did not come back to it. That was non-cyclic one. Now here it is cyclic photophosphorylation. Photo means light. Phosphorylation means preparation of ATP. So this is preparation of ATP from ADP and a inorganic phosphate to form ATP. ADP और पायरोफॉस्फेट मिला के जो ATP बनता है उसे फॉस्फोराइलेशन कहते हैं. अगर ये लाइट के प्रेजेंस में होता है then it is called photophosphorylation as in case of photosynthesis. Here only the PS1 takes part, not the 2 but just the PS1. Only PS1 takes part. And how it happens, say this is the chlorophyll, that is all the LHC here, and the antenna molecules and the reaction center. Here comes the light energy. Now this reaction center, it loses 2 electrons. Fine. It has lost 2 electrons which are accepted by primary acceptor. This is PS1. These 2 electrons, now they pass through the electron transport system. It passes through the electron transport system or electron transport chain, thereby forming ATP. तो यहाँ पे बन गया ATP from ADP and pyrophosphate. Then this electron, it comes back to the original source. So it has again come back to its original source. This is P700. PS1, its chlorophyll, its reaction center has absorption max at P700. So one question is that where is the enzyme for this cyclic photophosphorylation is present? I repeat, where is the enzyme for this cyclic photophosphorylation is present? Before that, just see here that in case of cyclic photophosphorylation, only ATP is formed. The reduction of NADP does not take place, so no NADPH is there. NADPH is not formed. Fine. Now where is the enzyme present? See, these are the different thylakoids arranged into grana, and then there are these stroma lamellae. Now, in these thylakoids, in the grana, you will find PS1 as well as PS2. I repeat, in this grana, you will find the PS1 complex as well as PS2 and the enzymes required for non-cyclic photophosphorylation. Whereas these stroma lamellae, one thing is they do not have PS2 because PS2 not required. So they are having no PS2 और एक चीज की उसे जरूरत नहीं पड़ेगी बच्चों. सोच के बताइए किस चीज की जरूरत नहीं पड़ेगी? उसे NADP रेडक्टेज की जरूरत नहीं पड़ेगी क्योंकि वो NADPH बना ही नहीं रहा है. एंड नो NADP reductase. These two things, they are not present in stroma lamellae. This cyclic photophosphorylation, it takes place in stroma lamellae. Clear? It takes place in stroma lamellae. And why and when this cyclic photophosphorylation occurs? That is one question ना. क्यों? किस समय होता है ये? नॉन साइक्लिक नहीं होके या अचानक से साइक्लिक क्यों होता है? This cyclic one occurs when the light wavelength above 680 nanometer is available to the plant. It also meets some ATP requirement that you will understand when you see the C3 cycle. So this was cyclic photophosphorylation. Now let us see the chemiosmotic hypothesis. This hypothesis, it explains how ATP is formed. What it is explaining? How ATP is formed. During photosynthesis, it will be formed in the chloroplast, in the thylakoid of the chloroplast, whereas during respiration also the ATP which are formed, it is formed by the mitochondria. Now, for the formation of ATP, what is necessary is the proton gradient. The necessity is that of proton gradient. What is proton gradient? Let us understand in an easy way. When we prepare the electricity, when we are storing the water in the dam, then we are letting it fall down, then the energy is generated, we convert it into electricity. Here, proton gradient means this is, see, this is thylakoid. The whole operation is going to take place inside the thylakoid. This is thylakoid. Now, what we are saying, that there will be proton gradient, means many hydrogen ions inside here, and in the stroma, this is thylakoid ना, तो thylakoid किसके अंदर है? ये thylakoid एक chloroplast के अंदर है. This chloroplast is having its stroma, and here these are the thylakoids. तो ये बाहर stroma है. So here it is present the stroma. So the hydrogen ion should be less in stroma and more inside the thylakoid here. Thylakoid lumen. This is thylakoid lumen. How this is formed, and when we broke down the gradient, जैसे ऊपर से पानी को release किया था, now we will allow the protons to come across this membrane with force, thus allowing the formation of ATP. It is as simple as that. Now, the participants here, let us see, this is the PS2. This is PS2. Now, it has to transfer its electrons to PS1, that you know, that Z or non-cyclic photophosphorylation. हम ये देख रहे हैं कि प्रोटॉन ग्रेडियंट यहाँ तैयार होता है तो कैसे होता है? Why the protons become more inside the thylakoid lumen? The first is that you know that during light reaction the water is split. तो जब water split होता है, and we have already seen कि water splitting complex, it is attached to the PS2 which is in the inner side of the, this is in the lumen. तो यहाँ पे वो water splitting complex है. तो ये क्या करेगा? It will split the water. When it splits water, then hydrogen ion is formed. Oxygen is also formed. That is waste, that will be released. Electrons are also formed. These electrons, this PS2 will take, because PS2 is going to lose its electron, has lost its electron actually to PS1. तो बचे कौन? ये hydrogen. So every time water is split, then the hydrogen, this proton, this will go on accumulating in the lumen. आसान है इतना? यही है पहला point. The first one, the first reason is the splitting of water. And only because this complex was present in the lumen itself, that's why the protons have been released in the lumen. ये पहला हो गया. Now here there is cytochrome B6F, and this side it is plastoquinone. This is plastoquinone, and this is not here, and this is plastocyanin. This is plastocyanin. Now the second step, why proton get more inside the lumen? This PS2, when it's LHC, it releases the electron. It is accepted by primary acceptor, which is present towards the outer side of the membrane, and when it transfers its electron, अब वो इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम शुरू होगी बच्चों। तो वो जो पहले कैरियर को अपना इलेक्ट्रॉन देता है, तो इसकी कंडीशन एक है, वो जो कैरियर को देता है इट इज नॉट एक्चुअली इलेक्ट्रॉन कैरियर, इट इज अ हाइड्रोजन कैरियर। वो कहता है ठीक है मैं आपका इलेक्ट्रॉन ट्रांसफर कर दूंगा, लेकिन मैं अपना हाइड्रोजन भी कैरी करूंगा। सो इट इज कैरिंग हाइड्रोजन एंड इट इज ऑल्सो कैरिंग इलेक्ट्रॉन। तो इलेक्ट्रॉन तो उसका फिर व्हेन इट गिव्स द इलेक्ट्रॉन टू द नेक्स्ट कैरियर, तो ये यहाँ से कैरियर से होते हुए तो ये यहाँ से अपने रास्ते ये निकल ही जाएंगे, ये इलेक्ट्रॉन ऐसे निकल ही जाएगा। हियर देयर इज FD एंड हियर इट इज FNR। तो इलेक्ट्रॉन इस रास्ते निकल जाना है, लेकिन जो हाइड्रोजन है it gets released into the lumen. So every time one electron is passed, then one hydrogen is released into the lumen. So the second one is the hydrogen carrier carries H plus and also electron. उसकी कंडीशन की वजह से वो हाइड्रोजन आयन को भी लेके आता है, तो हाइड्रोजन को अंदर रिलीज करेगा, इलेक्ट्रॉन को अपने रास्ते निकल जाने देगा। नाउ द थर्ड, नाउ व्हाट हैपन्स, अब यहाँ पे NAD है, NADP। नाउ दिस NADP is to be converted into NADP is to be converted into NADPH plus H plus. This reaction also needs this electron, तो ज़रूर चाहिए उसे, at the same time it needs protons also, and these protons it takes from the stroma. ये NADP reductase enzyme is present in the stroma, that's why this is happening. So the third one is the NADP reductase enzyme। NADP को reduce होने के लिए हाइड्रोजन भी चाहिए, तो NADP reductase enzyme is present in stroma। तो वो स्ट्रोमा से लेता है प्रोटॉन। इससे क्या होगा, स्ट्रोमा में प्रोटॉन्स कम हो जाएंगे, यहाँ बढ़े या यहाँ कम हो गए, दैट मीन्स ग्रेडियंट यहाँ अंदर ज़्यादा हो गया। वंस दिस ऑकर्स, इसमें बहुत सारे जब तक प्रोटॉन जमा हो जाएंगे, नाउ वी नीड टू ब्रेक दिस प्रोटॉन ग्रेडियंट। पहला चैलेंज है प्रोटॉन्स को जमा करना, दूसरा चैलेंज है इस ग्रेडियंट को तोड़ना। उसको तोड़ने का काम अब करेंगे। देयर इज वन एंजाइम ATP सिंथेज। दिस ATP सिंथेज इट हैज़ टू कंपोनेंट्स। एक तो है ये ट्रांस मेम्ब्रेन चैनल है, यहाँ पे दिस इज द CF0। ये एक चैनल बनाता है कि प्रोटॉन्स यू ऑल कैन कम थ्रू दिस पार्ट एंड यू कैन मूव आउट। एंड द अनदर वन, this is the CF1. This is CF1. So this is CF0, this is CF1. Through this CF0, these protons as well as these protons formed by splitting of water, these protons which has come from the stroma, these now move through this CF0, so that the energy which is generated, it is sufficient to change the structure of the CF1. Conformational change is there, which converts this ADP plus this pyrophosphate into ATP. That's it. This what we have written. This is the development of proton gradient, and here what we have shown, it is the breaking of the proton gradient. तो जब proton gradient break होगा तब ATP बनेगा. That means ATP formation, there are two steps. First, the formation of proton gradient because of three reasons, and then breaking of the proton gradient through this CF0, then this CF1, it produces the ATP. This ATP बच्चों and this NADPH, these two are the energy rich molecules required for the dark reaction, and they will be immediately used in the dark reaction for the formation of glucose. How? That we will see tomorrow, the concluding part of photosynthesis. Hopefully you have liked the video. Then please like, subscribe and hit the bell icon to get all latest updates.